Welcome everyone to our webinar, Introduction to Lifecycle Impacts and Exposure Assessment for Alternative Analysis. The objective of this webinar is to provide you a high-level overview of lifecycle impacts and exposure assessment so that you're all well prepared for the two-day workshop on August 9th and 10th to be held at the DTSC building in Sacramento, in California. My name is Sang-Won Soo, and I have been working on lifecycle assessment over the last 24 years. I'm a professor at the Brand School of Environmental Science and Management at the University of California, Santa Barbara. I'm also the director of the Chemical Lifecycle Collaborative Program here at the UCSB. I will talk about lifecycle impacts part, and my colleague Arturo Keller will talk about the exposure assessment part. Let me start with this picture. This picture evoked a lot of criticism to our former president, Barack Obama. Why do you think that this photo created such a criticism? Yes, it is in his hands, the plastic bags. Our president using plastic bags somehow created a lot of criticism, especially from the environmental groups. And what those environmental groups were thinking of was something like this, that plastic bag ends up here. What part of the life cycle of this plastic bag are these people concerned about? Obviously, this is the end of life phase. Once this plastic bag is disposed of, some of them may end up in the ocean, and it may adversely affect the ecosystem health. It may choke turtles, or it may intervene dolphins, as is shown in this picture. So that's what environmental groups were thinking of, and then they thought that, well, use of plastic bag is a bad thing for the environment, and it's a shame that our president is using not only one bag of plastics, but two bags of plastics at the same time. But then what about the alternatives, the paper bag? Is that better for the environment? Well, from a life cycle point of view, is not necessarily so. So life cycle assessment studies repeatedly have shown that paper bags use more energy, it uses more water, it generates more greenhouse gas emissions throughout its life cycle. So here comes the recognition that the environmental impacts are materialized not only by one specific life cycle segments of a product system, but throughout its life cycle, including resource extraction and refining, manufacturing, packaging and distribution, use, and end of life or disposal. Going back to the picture that I have shown to you, so the question becomes, is the use of plastic bags bad for the environment? And from a life cycle point of view, well, it is not necessarily so. It depends on which life cycle segments we are talking about. Is that the end of life phase are we talking about? Or is that resource extraction, manufacturing, use, and transportation altogether? So that's one question. Another question is, which environmental impact are we talking about? Are we talking about the ecosystem health impact, or are we talking about the toxic substances and associated impacts to the ecosystem and human health? Or are we talking about greenhouse gas emissions, water consumption, energy consumption? All depends on those two questions. Similarly, zero-emission vehicle, for example, is not necessarily zero emission from a life cycle thinking point of view. Zero-emission vehicles definitely generate no or very little tailpipe emissions. However, the use of electricity, the generation of electricity that the zero-emission vehicles needs to operate, they do generate greenhouse gas emissions, and other pollutants. So, for example, a recent study that we just published for the UN environment shows that actually battery electric vehicles are not only generating emissions, but also it may generate more greenhouse gas emissions than the gasoline cars. So depending on the background grid electricity, for example, if those battery electric vehicles are deployed in the areas with heavy reliance on coal electricity, for example, India and China, the greenhouse gas emissions from the use of the battery electric vehicles, actually life cycle of the battery electric vehicles might be larger or higher than the gasoline cars. So that's life cycle thinking in general. But what about life cycle thinking in the context of alternative analysis? So it goes in the same way. So which phase of the life cycle segments the chemical that we are looking into and evaluating the alternatives of contributing the most to the environment? Is that the production phase? Is that the use phase? Or is that the end of life phase? Or is that the distribution phase? And what cost to the environment does the chemical or the alternatives needs to be produced and used? Is that the energy and greenhouse gas emissions? Or is that water consumption? Or is that waste? Sometimes one chemical or alternative to that may be better for one area of life cycle segments or one environmental impact categories, but it may be worse for other life cycle segments or life cycle impacts. In that case, it becomes a trade-off situation and it becomes an issue of a judgment call. This is an example provided by the Alternative Analysis Guide by DTSC. So what this graph is showing is a comparison of greenhouse gas emissions per a functional unit, which I will explain later, between the priority products and its alternative. What you can see here is that for raw material processing, alternative produces more greenhouse gas emissions than the priority product, but for manufacturing, the opposite is true. So in the end, if it is just greenhouse gas emissions or CO2 emissions, then we can just all aggregate them and see which one contributes more to the greenhouse gas emissions, and we can think about finding a better alternative in that way. But obviously, if we are considering some other impacts than the climate change, the result may differ. This is another example provided by the AA guides by DTSC. What is shown here is the list of alternatives and the priority products, and these columns are life cycle stages: raw materials extraction, intermediate manufacturing, packaging, use, and end of life. And the impacts are shown as a low, medium, high scale. In this case, as you can see, the alternative D seems to be doing pretty well in all areas of human health and also air quality. And there are other impact categories that is not shown here, it's truncated here, but the impact on the water quality and other impacts can be also considered. So in this way, we can see what are the trade-offs among different alternatives and among different life cycle phases and impact categories, and that can help identifying the best alternatives at hand. So we briefly mentioned functional units. So what is a functional unit? So functional unit is quantifiable unit of main functions of a product. It is basically the performance metric. And it is used as the basis for comparison or the reference unit across alternatives. I will explain what I mean by that. So let's think about life cycle thinking or life cycle impact of two chemicals, methylene chloride and benzyl alcohol. Both of them are used widely as paint strippers. It turns out that from a life cycle assessment study, benzyl alcohol produces 0.79 kilograms of CO2 equivalent greenhouse gas emissions per kilogram. On the other hand, methylene chloride produces 0.67 kilograms of CO2 equivalent greenhouse gas emissions throughout its life cycle per kilogram. So based on this comparison, one can say that, well, methylene chloride produces less greenhouse gas emissions. Therefore, from a climate change point of view, methylene chloride is preferred. However, methylene chloride is less effective paint stripper as compared to benzyl alcohol if we compare kilogram per kilogram. So we need 1.2 kilograms of methylene chloride in order to provide the same functionality as compared to 1 kilogram of benzyl alcohol. So if we factor that in, then for the same functional unit, which is stripping the same area of wall, say stripping the paint on the same area of wall, the methylene chloride will produce 0.82 kilogram of CO2, whereas benzyl alcohol produces 0.79. So the result can be reversed if we factor in the functional equivalency of those two chemicals. Now let's let's move on to the life cycle assessment. So life cycle assessment is a tool. So life cycle thinking was more a conceptual framework, and life cycle assessment is basically the quantitative tool that support life cycle thinking. So life cycle assessment is a method to quantify environmental impacts of a product system throughout its life cycle. So let's talk about the 4 phases of an LCA. What is shown here is from the ISO standard, ISO 14040. Life cycle assessment has four phases: goal and scope definition, inventory analysis, impact assessment, and interpretation. And as you can see, the arrows going back and forth. This is not the sequential process. This is iterative process. So we need to start with goal and scope definition, and then inventory analysis. And we may want to go back to goal and scope definition if the inventory analysis cannot be performed as planned. And throughout those processes, interpretation need to be performed in each and every step. And once it is finished, it can be applied to the product development, marketing, eco-labeling, public policy making, and regulatory purposes like what we are talking about here today. So the first step is the goal and scope definition. And in a nutshell, goal and scope definition is all about planning. So it is about the planning the entire life cycle assessment study, defining the objectives, system boundaries, functional units, impact assessment methods, and also interpretation methods. The second phase, life cycle inventory analysis, is about, in a nutshell, data collection. There are 2 types of data that life cycle inventory is concerned. One type of data is inputs and outputs from and to other unit processes. So other unit processes might be, for example, power plant that produces electricity to my plant, and it can be the products that I'm producing in support of other companies, other clients. So those are the inputs and outputs that are generally manufactured or generally um, produced as a result of anthropogenic activities, economic activities. That is what is called intermediate flows in life cycle assessment. So there is some kind of human intervention or human transformation happening for those flows. Whereas the second set of data that we are collecting in life cycle inventory phase is inputs from and outputs to the nature. So this the examples could be crude oil extracted from the oil field, or CO2 emissions liberated from the stack to the air. And there will not be any further human transformation or prior human transformation to those flows. And those flows are called elementary flows. Basically, those are the interactions of the anthropogenic systems with the nature. That's what is called elementary flow. So that's what we are collecting in life cycle inventory analysis. And those flows are the basis upon which the impacts are quantified in the next phase. So during the life cycle inventory phase, we generally draw what is called process flow diagram. And that process flow diagram shows what are the inputs and outputs from other product systems or the nature, from and to the nature. So those are the elementary flows that we just talked about. And if it is coming from other man-made system, those are product flows or service flows. They are shown here. Those are intermediate flows. And the system boundary of my product may contain multiple unit processes. I will show you one example of lifecycle inventory. I just visited the US LCI website by NREL, National Renewable Energy Laboratory. And it looks like this, and it contains, as you can see, 5,224 record. And I just clicked the first one, acetic acid at plant. I truncated to fit into the screen. What it shows is the outputs, and also later on it shows also inputs. And so outputs includes the product itself. So it is normalized by 1 kilogram of the product. And in order to produce that much of product, acetic acid, it produces so much of unspecified acid, so much of ammonia, to water, to air, and carbon monoxide to air, etc. So these are the emissions generated throughout the life cycle of acetic acid for 1 kilogram. So this is how life cycle inventory looks like. Once the life cycle inventory is compiled, the next step is life cycle impact assessment. And life cycle impact assessment is basically aggregating the life cycle inventory flows into impact scores. What is the contribution of those life cycle inventory flows, elementary flows especially, to climate change or to human toxicity or to energy consumption? That's what life cycle impact assessment is concerned about. There is a little bit of complexity in life cycle impact assessment. I will not be able to go into too much of detail during this introductory webinar, but we will have more opportunity to talk about in more detail during the workshop. There are 3 steps in life cycle impact assessment we generally distinguish. They are characterization, normalization, and weighting. And we will focus more on the characterization part. And there are 2 different types of characterization method widely discussed. One is called midpoint, and the other one is endpoint. I will show you what we are talking about here. But first, let's look into the most widely used characterization methods. So characterization is really about aggregating life cycle inventory flows into quantifiable, commensurate environmental impacts, such as climate change. And the most well-known example of which is global warming potential, especially GWP 100, which is based on 100 years of time, the radiative forcing during that 100 years of life, 100 years of time frame. So carbon dioxide, CO2, is used as the reference flow. In this case, it is the reference of all other greenhouse gases. So everything is normalized by the radiative forcing within 100 years of time frame of carbon dioxide. Therefore, carbon dioxide has GWP of any kind as 1, and then the radiative forcing of other greenhouse gas emissions, greenhouse gases, are measured relative to that of carbon dioxide. So in this case, according to the assessment report 5 by IPCC, in the last column, you can see that methane has 28 times of radiative forcing within 100 years of time frame as compared to carbon dioxide, and that can be used as a factor to convert kilograms of methane into kilograms of carbon dioxide equivalency. That is what is called, in a nutshell, characterization. But there are all kinds of other environmental impacts, and those environmental impacts are materialized through very complex environmental pathways, or according to the ISO standard, is called environmental mechanism. So the elementary flows are shown on the far left: CO2, HCFC22, N2O. Those are the elementary flows, and they affect the environment through multiple environmental mechanisms or pathways. In the end, it will affect human health, ecosystem health, and resources that you are seeing on the far right. But in between, it will do some other things before it can be materialized as a human health impact or ecosystem health impact or resource impact. So for example, for CO2, what it will do first is to increase the radiative forcing. And that may increase the global mean temperature, which in turn may affect the sea level rises or changes in precipitation patterns, or it changes the habitats to the ecosystem. So in between, there are multiple different ways in which those emissions affect the environment and the ecosystem. It's very complex. So no way we understand all these complex mechanisms, but science help us to understand at least some of them, and those understandings are distilled into what is called characterization factors. And those characterization factors, therefore, are not perfect, but that they reflect some of the best sciences that we have access to. So the midpoint characterization is really about measuring those impacts at this level before those impacts are all materialized toward the end of this whole environmental mechanism. And endpoint indicators or endpoint characterization methods are focusing on toward the far right side of the environmental mechanism. And there are cons and pros in both approaches. We'll talk probably more about this during the workshop. These are the examples of midpoint indicators or midpoint characterization, the impact categories that characterization addresses. So, for example, acidification or eutrophication, smoke formation, these are all distinguishable environmental impact for which a set of characterization factors have been developed. So the last phase of the life cycle assessment is the interpretation. The the block that you have seen on the far right of the diagram. What interpretation phase is really about is to identify the significant issues. The significant issues can be lifecycle inventory or lifecycle impact assessment result. It can be certain impact categories, or it can be a lifecycle segment, or it can be certain emissions or resources that are consumed by the lifecycle of the products or chemicals, let's say. And then what the interpretation phase really does is the evaluation of the result in terms of its completeness, sensitivity, and consistency. And if the level of completeness or level of consistency does not meet the initial requirements set by the goal and scope definition, we will have to go back to the goal and scope definition and either revise the goal and scope definition and lower the bar, or we need to collect better data to meet the requirement. Finally, the interpretation phase is the phase where the conclusions are drawn and recommendations are drawn and reports are written. So that concludes my section, and I would like to hand it over to my colleague Arturo Keller for exposure assessment.
Next, I'm going to talk about exposure and risk assessment and how they can be used for alternatives assessment. My name is Arturo Keller, and I'm a professor here at the University of California in Santa Barbara. So exposure and risk assessment go hand in hand. For you to do a risk assessment, first you have to determine the exposure, and that is very useful for us to use in alternatives assessment, which also requires for us to understand what are the various exposures that someone may have. So we have a chemical, and that chemical is used in some activities. There's some release either directly to the person who's using it, and it may be, for example, a dermal release or an inhalation release. You breathe it, it's on your skin, or you may ingest it. And the release also results in some transport, which then results in exposure or potentially more people and also organisms. Then we have to compare that to the toxicity to be able to understand whether that exposure is going to cause some injury or some issues. So I'm going to walk you through what are some of the key concepts, the basic concepts in exposure and risk assessment, and talk briefly about how they may be used for alternatives assessment in preparation for the workshop that we're going to have later on in August. So the talk will cover the human and ecological health risk assessment. What are some of the basic steps for doing a risk assessment? And again, how that may connect later on with the alternatives assessment, how we determine toxicity, and there's various types of toxicity. There's non-carcinogenic toxicity. This may be something that causes some form of illness, or it may cause eventually death. There's carcinogenic toxicity. And there also, we have ecological toxicity or ecotoxicity. We're also going to talk about how do we estimate exposure and how we go about all the different things that go into an exposure assessment. And also, we'll talk about risk characterization briefly, what that is and how that may be used for your alternatives assessment. So first, before we go too far, risk assessment is not alternatives assessment. Alternatives assessment may use risk assessment as one of the tools to help you with your assessment of the alternatives, but there are important differences between these two, and I want to highlight those differences. So risk assessment is really looking at what's an acceptable level of harm. For example, the number of cancers that may be increased due to exposure to the chemical per million people, and is it above or below an acceptable level? Does an activity or product use fall within the acceptable level? And the activities usually are typically considered in isolation. And so that's somewhat of a difference between that and alternatives assessment, which is really looking more as, is this potentially activity or product necessary, potentially hazardous activity or product necessary? How can the hazard be reduced or eliminated? What other options are available? And typically, it will consider multiple activities in the same alternatives assessment. Also importantly, alternatives assessment looks at many other factors that are not just the toxicity. So it will be looking at the things that Dr. Su mentioned before, whereas risk assessment usually is focused more on the toxicity of the chemical in the product. So some of the basic steps in risk assessment. First, we have to identify what are the hazards, what are the chemicals that may present an issue. So for example, here I have a paint stripper, and we may have different chemicals in that paint stripper. And so those are the hazards that are identified in that product. Then we go into the exposure assessment. How are people or organisms in the environment exposed to the chemical of concern? And then we go into a dose response assessment. For many chemicals, this has already been done. For some of them, this needs to be developed, and that's a step that's important for risk assessment. Understanding, does it cause cancer, or does it cause nausea or coma? And then putting together the dose response and the exposure assessment, we go and characterize the risk. And as shown here, we can characterize it from something that's rare and has a minor severity, has very low risk, all the way to something that is very likely and would have catastrophic risk, which would then have an extreme risk. So we can sort of have a gradient of risk and then draw the line at some point and say, this is what's acceptable for a risk. To put it into more quantitative terms, when we talk about, for example, hazard identification, what we're really saying is, let's say here I have a situation where we have an underground storage tank that has leaked a number of chemicals in the gasoline. And so we have benzene, toluene, xylenes, MTBE, and pentane at different concentrations. So we've identified the chemicals and the concentrations at which they may be present within the product. Then we go into the exposure assessment, which looks at what's the route of exposure. So this chemical is spilled underground. How are people likely to be exposed? So it may be a construction worker or someone participating in the cleanup activities. It could be someone drinking the groundwater that's contaminated, or it could be children playing in the soils that have been excavated or that were left behind after the cleanup activity. So these are basically different routes of exposure. And then we also get an idea of what are the concentrations and the dose that these people may be exposed to. And the reason for that is that toxicity is all based on dose, toxicity assessment. We need to know what is the dose that causes a response. And I'll talk more about what we mean by response, but basically what's the dose that causes some harm or injury in terms of the toxic effects. And so then we put all those together, and when we can characterize the risk and say, well, these are risks from these chemicals, and they're due to different routes of ingestion or routes of exposure, and try to get an idea of whether this is above or below a particular threshold of acceptable level. So to determine toxicity, we first have to do studies with organisms. Most of these are done that way. Some of them are done with epidemiological studies, so people looking at what is the situation where people are exposed in the workplace, for example, to these chemicals, and from there gather that information. But many of these are done as basically animal studies. And what we're trying to figure out is, does the exposure cause an increase in incidence of an adverse health effect, whether it be in humans or it would be in, for example, rats or mice or frogs. And then a dose response assessment, a quantitative dose response relationship. So we have exposure and concentrations, and for example, shown here is the number of organisms that are still healthy or that have some effect, and we can see at some point we begin to detect some effects, and so we no longer have all our population in the healthy levels. So some definitions, some important vocabulary. We have the NOAEL, which is the no observable adverse effects level. We have the LOAEL, the lowest observable adverse effects level. We have the EC50, which is a concentration at which 50% of the exposed organisms experience effects. And then for cancer, we talk about a slope factor, sometimes called the cancer slope factor, and is the cancer risk per unit of dose. So it tells you, what's the increase in risk as you increase a unit of dose. So if you increase the dose by 1 milligram, what is the increase in the cancer for that population? For non-carcinogens, or when we're looking at just the non-carcinogenic risk. We do a toxicity main assessment, and what we find is these different measures of acceptability. One of them is so-called the reference dose, a lot of times also mentioned as the RFD. And it's the maximum amount of a chemical that an organism can absorb without experiencing chronic health effects. Provides an estimate of continuous daily exposure for the general population, including sensitive groups, without an appreciable deleterious effect. So how much can you be exposed without showing effects? The units are in milligrams per kilogram of body weight per day. And that helps us to be able to adjust it to the weight of different humans, for example, children versus adults, females versus males. Also, when we're taking data from, for example, mice or rats to humans, it helps us to make that translation between the exposure for those organisms, which have a very different body weight than what we may have. And it includes very predictive assumptions. So it's not something where we just take the direct result from the animal study, but the people take some very protective assumptions to translate that into what would be the reference dose. The acceptable daily intake, the ADI, is also a similar measure, and what this does is uses the LOAEL, the lowest observable adverse effect level, to estimate what would be an acceptable daily intake. For carcinogens, we typically assume that there's no threshold level for carcinogenic effects. In other words, any exposure will have some effect. Now, it may be that it's very small, and so there's work that people do when they're doing these assessments, because a lot of times we don't see cancer until you reach a particular dose. But that doesn't mean that there wasn't the beginning of a cancer. By nature, these experiments are done on a short-term basis, and as we know, most cancers take time to develop. Exposure to a particular chemical, a particular injury, may take a while for it to represent observable tumors, observable effects. So we estimate the excess cancer per unit dose and use the slope of the curve to determine the slope factor, and the units are also in risks per milligram per kilogram per day. So it's also based on the body weight. Again, very conservative assumptions to try to be as protective of the potentially exposed populations. And it's determined as the increased lifetime risk per unit of dose. Next, I'm going to talk a little bit about exposure assessment. So in exposure assessment, what we're trying to see is, given the use of these different chemicals, maybe in industry, or as you go and gas up at your local gas station, or other activities that you may be involved with where you have contact with the chemicals or the chemicals being used, the chemical eventually is released into the environment, and it may go, for example, here in this example, into the atmosphere, and wind moves it down gradient, and then there's dry and wet deposition. There's also some transformation of this chemical in the environment. And eventually that deposition and that movement gets it into our soils, into our waters, and then into our foods. And so there could be some bioaccumulation in the food that then basically goes into humans via ingestion of contaminated food, or it could be that you're swimming in a contaminated water body, and so it's by dermal absorption, or you could be breathing the air and it's by inhalation. So depending on the distance that there is between the release of the chemical and the point of exposure, the concentrations need to be calculated. And that's what we mean by exposure assessment. First, what are the pathways, the routes that the chemical may take from the point of use to the point of exposure? It may be very close if you're looking at occupational exposure. It may be very far away if you're talking about something that's, being used in industry and eventually gets out to people in further away, or to organisms in the environment. So one thing we need to do when we're doing exposure modeling is to think about, what does our environment look like? So here I have a schematic of what a typical environment may look like. We have an urban environment, and we may be looking at the situation within that urban environment. But typically, we may have more than just that. We have, for example, the atmosphere above it. And so that atmosphere contains not only gases, but also aerosols, small particles that can travel through the atmosphere and deposit and take our chemicals to other places. There's agriculture, and maybe, for example, it's a pesticide being applied in agriculture, and we want to see what the risk is to people in everywhere else. We also have undeveloped land that may or may not have exposure, but it may be something where we want to think about what's the ecological risk in some of those environments. We also have a wastewater treatment plant, and so many of our chemicals are used, and then we wash our hands, or they're excreted into the wastewater, and it goes to the wastewater treatment plant where it's processed, but many of these novel chemicals are not really the target of our traditional wastewater treatment, and so they go through the effluent into our fresh waters, or the sludges and biosolids that are coming from the wastewater may be applied to agriculture. And then that goes to freshwater. Freshwater may connect to the coastal environment, so we have a possibility, a pathway for it to go from the urban environment into freshwaters and into our coastal and marine environments. And we also look at our soils, not just the top surface, but also as we go deeper and deeper into the soil, as well as the sediments within our water bodies. So this is our conceptual model of what our environment looks like. And we have to think about what are the distances that are associated with, again, the point of use and release to the point of exposure, as well as what are the transport pathways and the mechanisms by which a chemical gets transported from the point of use or release to the point of exposure, and transformations that may be occurring. We're talking about human routes of exposure. We have, in the outdoors, we have inhalation via air or ingestion via food. In the indoors, we have inhalation mostly. We also have ingestion of the food, and that may be food that comes from the outside internally. And we also have dermal exposure as a possibility, where you're actually, for example, in this case, the middle case, you have paint that gets applied on your hands. That's a direct dermal exposure. It could also be that you've touched a surface that was contaminated with, so it's with a chemical, and that's an indirect exposure. And then we also have to think about the ecological exposures. And as I mentioned, as we think about the conceptual model of our environment, we have different places where exposure can occur for organisms: in freshwaters, in marine environments, in sediments, either the benthic environment as well as our soils. So there's different routes and points of exposure. So some of the major components in exposure assessment include the use of the chemical. What's the chemical and how it gets used? For example, here may be a personal care product, and you apply it on your hands, and from there, there's first the direct dermal contact, as well as you wash your hands, and so that takes it into our wastewater, and it's transported out into the wastewater treatment plant and eventually out into the environment. And then there's exposure to other people or other organisms to that chemical. When we talk about use, we can think about industrial uses, indoors or outdoors. In many places, we have control devices to try to minimize the exposure, but not everywhere. And sometimes those control activities, for example, using personal protective equipment, are bypassed. And so we see, for example, here in some of the examples, where some people are using their personal protective equipment quite well. In other cases, they have either none or they're not using it adequately. So there could be exposure indoors or outdoors. For consumer use, in many cases, consumers do not use personal protective equipment. And so there we have to think also about what is the product category. So for example, is this product a foodstuff? Is it a personal care product? Is it a paint or a coating? Is it a solvent that the user may be using, a pesticide? And then the functional uses. So we'll talk more about what functional uses are in the workshop. But basically what we mean is chemicals have uses, for example, as surfactants or as disinfectants, antimicrobials, solvents. And so those chemicals go into a product because of the function they provide within that product. And so that helps us to calculate the exposure. Then we have chemical release, and what we're trying to figure out is where and how much. So we have release from the production activities. So production activities may have some releases into the atmosphere, into their waters or wastewaters, or into soils. There's the use. So when people are participating in the activities that use the products where this chemical has been embedded in, and so that again has releases. And then there is disposal either to a landfill or to a wastewater treatment plant or a waste incineration plant. And so all of those eventually have connections to different parts of the environment. So what we're trying to show with this diagram is that there's a connection at every point in the life cycle of the chemical, from its production and embedding it into a product that people then use, and then into the various disposal pathways. And then eventually that may get it into different compartments in our environment. So release takes care of that. Once we have the release, then we model the fate and transport. So here we have, again, our environment. And now that we've released things into the environment, we go ahead and use different models to make predictions about what would be the concentrations. So for example, here we're looking at some concentrations in the right-hand side where you have the concentration of a chemical in the San Francisco Bay Area. In the top diagram, it shows air, and the lower figure, it shows fresh water. So we see different concentrations, and we see a lot of variability within a particular range of concentrations. And that has to do with, for example, atmospheric transport events. So there's high wind events, or there could be some rain events. There could be some other events that move it through the environment. And even though the release in this case was to the atmosphere, we also see it in our fresh waters and in our sediments as shown down below. So that's what we're trying to do, figure out what is from the release to what would be the concentrations at which people in the environment may be breathing that air or organisms in the fresh water may be exposed to, and what does that mean for them. So then we go into risk characterization. We evaluate the risks to the potentially exposed populations. Those could be human populations, or they could be organisms in the environment, again, in our freshwaters, in terrestrial environments, so it could be in agriculture, or it could be in a natural forest. And so it also could be marine environments. Risk characterization brings together the toxicity assessment. So what are the issues that we have when someone's exposed to a particular dose, and the exposure assessment. And therefore we get these quotients or indices. So a non-carcinogenic hazard quotient. So if it exceeds the quotient or the ratio by a factor of 1, then it's a higher risk. Or an index of carcinogenic risk, what's the increase in the cancers per, let's say, 1 million people that is due to the exposure to this chemical, or the ecological health risk. Is this going to be causing increased fish kills or increased damage to birds or other organisms? It's a qualitative or quantitative assessment of the uncertainty. We also have to do a quantitative or qualitative assessment of the uncertainty shared with each step of the risk assessment. So it's important to take into consideration the fact that we have a number of uncertainties, and that those have to be taken into account. For example, we know what the average person may be doing in terms of use of the product, but not everyone is going to be doing it at the average rate. And so what is the uncertainty associated with the use of the product? Or not everybody has the same body weight. What's the range of body weights? The time that you spend at the activity may vary significantly. What we know about the environment may also have uncertainty. So all of these things get taken into account so that we can then make a more informed decision about what would be the real risk if exposed to these chemicals in these concentrations. Mixtures of pollutants are handled by adding their individual risks, so we can take into account mixtures of pollutants. And there is some work that has been done to sort of try to see whether there are synergistic effects. In other words, do exposure to more than one chemical cause an increased effect more than just the addition of them? And so that is important. And then risk characterization basically it stops at this step where it says, okay, here is your risk. It's increased risk of non-carcinogenic hazard or carcinogenic hazard or ecological health. And then the next step is to do risk management. How do we reduce the exposure? How do we change things? So in the case of alternative assessment, one alternative would be, can we change the chemical that's providing that particular function and therefore have a lower risk? Or can we change the way we do a particular activity? For example, if you're doing a stripping paint from a wall, is there a better way for you to do that activity that doesn't require the use of those particular chemicals? And it may be a non-chemical option. So there are chemical options, changing the chemical, or there may be some non-chemical options to achieve the same objectives. And so that's what risk management is all about. Risk characterization leaves it at telling you, for example, this is an example with PCBs, a type of chemicals, polychlorinated biphenyls, and we did this case study in San Francisco Bay. And what we find is that for many of these environments, the risk is very low. We don't foresee a significant risk in those conditions. In 2 of them, we have no data, and it's because we don't know exactly what the risk is to organisms, the hazard is to organisms in sediments. for PCBs. And then on the right-hand side, we have another way of portraying the same type of information, showing different lines for another chemical, different lines, the NOAC, the LOAC, the different EC10, 50, and so on for different organisms. And we see, for example, for a particular nanopesticide, that it eventually reaches a threshold level, for example, the common carp, which is a concern. For others, it crosses a number of lines. So this particular chemical, this nanopesticide, would pose a risk if used in these environments to different organisms. So again, just to reiterate, there's a difference between risk assessment and alternatives assessment. We've gone through this, but I just want to make it clear that risk assessment is not alternatives assessment. It's a tool that can be used for alternatives assessment. It's very helpful in providing some of the important aspects that you need for your alternatives assessment, but alternatives assessment looks at many other activities and many other aspects of the alternatives that you may be considering. So please take that in consideration as you do your alternatives assessment. So I'd like to finish up by inviting you to this workshop that we're going to have August 9th and 10th. Is the Alternatives Analysis Workshop on Life Cycle Impacts and Exposure Assessment. Again, August 9th and 10th of 2018. It will be in Cal EPA headquarters, and you can go ahead and register for that, and please do so that we have an ability to accommodate you at the participation. So thank you very much for listening to this, and we'll hope to see you at the workshop and provide you with much more information about how we go about doing life cycle impacts and exposure assessment in support of alternatives analysis.